we are opening this one up to the public to tell people about Cosmos and some of the work that's been going on within the Cosmos project. But normally they're for members of the society only, and you can learn more about the society at our website, ascctalks.org. I also wanted to let you know that we're having an annual meeting, November 12th. It's our third annual meeting, and you can get information at our website, of course. There's going to be a couple of plenary lectures. The theme is adverse outcome pathways. And but we're hoping to bring in some basic researchers and other sort of outside the normal realm of AOP's thinking. So poster abstracts are due on September 26th, and you can see more about what the meeting will be about online. I'm the secretary of ASCCT, Christy Sullivan, and just to let you know a little bit about the webinar, it will be recorded, and everyone is on mute right now to cut down on the background noise. You will be able to access the recording. Members can access it through the password protected member site. If you're not a member, I encourage you to become one. Or if you are listening and you want to access a recording of the webinar later, please make sure to email me because this webinar doesn't save your email addresses or anything like that. Unless, of course, you emailed me directly for the access information, because in that case, I will already have your email address. So today's webinar is about Threshold of Toxicological Concern. It's titled An Approach for Safety Assessment and its Applicability to Cosmetics-Related Chemicals. And it's going to be given by Dr. Chihae Young. Chihae is the CEO of Molecular Networks and the Chief Scientific Officer of Altamira and an adjunct professor at The Ohio State University. She was previously the Chief Scientific Officer of LeadScope before spending 3 years at FDA to learn about risk assessment issues. Her contributions to the field of computational toxicology include the ToxML data standard, QSAR prediction methods based on structural features, and development of ToxPrint and Chemotyper. Through this long 15-year relationship with FDA, she enabled implementation of computational methods and the SERIES system at CIFSAM. This collaboration includes the Threshold of Toxicological Concern. In the Cosmos project, she leads the TTC work package, and her company Altamira developed the Cosmos database. She also served as one of the first scientific board members of the ASCCT. So we're very happy to have Chihae giving this presentation. First, we're going to have Andrea Richards, who is a project coordinator for Cosmos, give a little quick review of the Cosmos project itself. So let me give Andrea the screen. And Andrea, you should be unmuted.
All right. Hello, good morning or good afternoon, everybody from sunny Liverpool. And I hope you're able now to see my screen.
Yep, there we go.
Okay. So I basically would like to shortly set off the context of the TTC work presented in the following and introduce the Cosmos project. So the work is done in the context of the cosmetics, or was started with the cosmetics regulation in Europe. In the European Union, animal testing has been completely banned for safety assessment of cosmetics. Sorry, I think you still see the. So last year was the final, or the full ban came into force, which meant complete banning of animal testing and marketing of products which contain ingredients tested on animals for all endpoints. But obviously, safety assessment of products for the consumers is still very important, and so alternatives had to be found to guarantee safety assessment. For complex systemic endpoints, especially, these alternatives were not available yet. So the European Commission, together with the European Cosmetics Trade Association, funded a cluster of research projects to investigate and find solutions for the safety assessment for cosmetics ingredients. And this cluster's initiative is called SURAT-1. SURAT as such is an initiative for the safety evaluation, ultimately replacing animal testing, and SURAT-1 is the first phase based on the replacement or towards the replacement of in vivo repeated dose systemic toxicity. The SURAT-1 cluster comprises 7 projects. The funding is in total of 50 million, both from the European Commission and Cosmetics Europe, and the project period is 5 years, start in 2011. And one of the projects is Cosmos, as you can see here. Cosmos aims to use computational methods to predict the effects of repeated dose exposure to cosmetics. And Cosmos has 15 partners in total, and 2 of them are in the US. Altamira is one of them, and US FDA, another one. And the Cosmos project comprises 4 main areas of research. The first is to create new toxicological databases, especially for cosmetics-related chemicals, obviously. And the database has been released. The version 1 has been released last December. It's publicly available, as you can see from the link shown on the slide. There was a webinar about this database in February, and you can access more information and the recording of the webinar through the link to this website shown on the slide as well. Another area of the Cosmos project is the research into threshold of toxicological concern, obviously, to evaluate the extension to cosmetics-related chemicals, and that's what Chihae Yang will talk about in this webinar. Another area is developing in silico models, grouping approaches mostly, which are set within the framework of adverse outcome pathways, also molecular modeling, and important as well, toxicokinetics models, PBPK, and in vitro to in vivo extrapolation. But we are not going into these details now. If you would like to get more information about these areas and the Cosmos project, you can access our website or contact us, obviously, with questions, etc. But for now, I would like to give Chihae the microphone to present the actual work on TTC. Thank you.
Okay, thank you, Andrea. And Chihae, I'm going to give you the floor and unmute you here. Okay.
You unmute.
Yep, you are off mute.
Okay.
We can hear you.
Well, thank you very much, Christy and Andrea, for inviting me here, and Christy's introduction. So we'll get into this describing the TTC work that we have been doing at Cosmos. So first, we would like to actually briefly go over the history of the TTC, and then defining what the chemical space of the cosmetics-related products may be, and then applying that to TTC approach and where we're going from there. So I'm at the slide of the first introducing threshold of toxicological concern. I've noticed that many people who are in the audience might know better than myself on this topic. And this is actually one of the very pragmatic and useful methods when we actually finally agree on and implement. So I'm glad we are having this opportunity. The concept of TTC has been pioneered by FDA scientists over several decades, starting from Frawley back in 1967, and the leadership of Rulis in 1980s and 90s, and then Cheeseman took the baton and further expanded that to the CPDB-related cancer TTC endpoints, and we will go through that a little more detail in the later. This concept of threshold is actually very familiar aspect that originated from our old, the famous 15th century physician Paracelsus, that dose makes the poison. So we could assume that there may be a dose below which no relevant effects are expected, regardless of the substances, and that these concepts may be applicable for substances used at low amount or low exposure, and when we don't have enough data, with limited toxicity data available. So we're back to this threshold of regulation exemptions that we call TOR. It is a special case of the TTC concept, and it has been implemented in CFSEN to regulate the part of the program as the food contact materials. So this actually provides the estimated consumer exposure threshold at 0.5 ppb, and any exposure under the 0.5 ppb would be considered not expected to have appreciable human risk. If, so the exposure needs to be below 0.5 ppb, and also the compound, the substance, should have no evidence of carcinogenicity in humans and animals, and there are no structural basis for suspecting it be a carcinogen or potent toxin, so structural alerts. And this morning, actually, Kirk Artisan sent me one of the example of the first TOR candidate. It was a tetrasodium EDTA in 1996, and it's a so it has been a very useful and effective way of one of the ways to regulate the food contact materials in FDA. And the next, please.
Okay.
So related to this, that it's not surprising that FDA has led the first cancer TTC activities and in the initiatives. And it's actually related to the Delaney Clause back in 1958. Again, we mentioned before that the materials must not be known carcinogen to humans or animals. So in 1999, Mitch Cheeseman's publication, the authors analyzed the data set based on the RTEX and PAPA and the carcinogenicity potency database, and compared between the cancer endpoints and the target organ endpoints and their sensitivity, and they have concluded that the carcinogenicity is the endpoint of most concern and sensitive at the lowest dietary concentrations. So ever since then, this cancer potency database has been used to sort of as a database source for this cancer TTC approach, and using the point of departure of the TD50 values, which is defined as the dose that induces tumors in 50% of the animals in their lifespan, normal lifespan. And the initial compound counts were 709, and it has a long history of relationship between the lowest scores of carcinogenicity potential, the carcinogenicity potency database and FDA. And it also is the first time along with the Monroe's approach, that you start using the structural alerts to actually separate the groups. For example, very highly carcinogenic or known toxic groups, such as N-nitroso, phosphorus, and the compound classes like this. And the threshold was actually established by a risk management paradigm to come up with the virtual safe dose by applying this cancer risk of 1 in a million cancer risk. So it is definitely different from the non-cancer TTC that we'll talk about in the next slide.
An answer.
Can you go to the next slide? See? Thank you. In parallel with the FDA's cancer approach, because they consider that as the most sensitive, and they have for that because of the limit clause. The other, more generic TTC has been actually initiated, and Monroe and others had publication back in 1996, and applying this TTC concept in general toxicity. So he used mostly chronic toxicity experiments, and the data were gathered from the EPA, IRIS, and the JEFA, and the literature, and so forth. And the point of departure was no observed effects level he used for the chronic, and so the subchronic and other studies were converted. And the first database had 613 substances, and it's the first and the last. We still use this database. And then he applied to group the compounds into 3 classes by using the 1978 publication of the Kramer and others, and came up with 3 groups. For each group, defined a 5th percentile threshold. And so the next slide, I would like to show you a little bit more about the Kramer classes. So you could actually see this pattern emerging that whether it's a cancer or generic TTC, non-cancer or cancer, there are 2 paradigms we have to handle. One is toxicity data and its POD, point of departure method, and the other is chemistry, the chemical groups, how we categorize those. So the Kramer decision tree was first proposed for the fragrances and food additives, and it's a decision tree type of method, and they group into 3 classes. The first class being the low toxicity, which he calls innocuous, and it's the simple structures with efficient metabolism. And the class 2 is less innocuous, and it does not contain any structural features suggesting toxicity. The class 3 then being the least innocuous and strongly presumed to be not to be safe, or it may even suggest toxicity or have reactive functional groups. So this 1, 2, 3, he grouped the compounds this way. They actually had about 81 compounds in the training set and looked at those when they developed these classifications. And so, for example, the first one up on the top, there was that's the compound containing nitroso, azo triazono, that's containing quad, and they remove the compound and then they go down the food chain. So let's go back down to the next slide. Okay. So what I'm actually trying to share here with you is that Kramer in 1978, he published with its 81 compounds of class 1, 2, 3, and each class had a no effect level, a range. Okay, the class 1 has about 50 to 254, class 2 has about 5 to 200, class 3 from 0.03 to 500. So you could see how this distribution is slightly moving from the class 1, 2, 3, the innocuous, to the most potentially toxic, towards the lower end, towards the low dose levels. Then in Monroe, he used the same classification. I am not done yet. Thank you.
Sorry.
Applied to 613 compounds, and you could see this NOAEL ranges of each class. So it widens the class of 1, 2, 3, And from this distribution, Monroe and others derived the threshold for each Kremer classes. So this classification of the compounds and correlating with the no observed effect levels of toxicity has been known from 1978. Monroe just is published with a larger database. Can we go on next? So now we talked about two cancer and non-cancer TTC approaches. And in this 2004, Cruz and others, they combined this into one framework. So the yellow top is actually the cancer part. And the green in the bottom is the non-cancer part. The cancer part is actually, as we talked about, cancer endpoints are much more sensitive. So we have the estimated daily intake threshold about 1.5 microgram per day. Now, if it doesn't belong to the cancer part, then we go into the next one and say, addressing this non-cancer part. May I go back?
I'm sorry. Chihae, I just realized that I can give you control of the mouse.
All right.
So you should be able to advance the slides now.
Let me see that I could do this. Yes, it moves. Thank you. So continuing on the Monroe story, that the threshold of each of the Kremer classes, class 1 and 2 and 3, the first one is actually calculated from the 5th percentile, the animal dose level, that's the NOAEL, the 5th percentile. Then it was calculated to the human exposure by the daily intake was NOAEL divided by a safety factor of 100, and then we multiply by 60 kilogram per as a human adult body. So this is a conversion. That's how we just calculate it. Oh, I don't seem to be able to go to the next slide. Oh, okay, here we go. Okay. So what I like to do at this point is taking a very simple example and go through this decision tree. And I have to apologize that it was difficult to come up with a good example that it is not politically sensitive. So because it could be, some of your compounds or some maybe you're interested. So this example may not be really pertinent, but let's just assume that what I wrote here as just assume that it was a fact and we move on. So let's say this 2,6-xylenol compound is that we are interested. And then first you look around the data, and first the carcinogenicity data, there are not good enough carcinogenicity data that I could find on this 2,6-xylenol. However, there are some data reported in 2,4 and 2,5-xylenol in skin tumor and dermal studies, and also I'm not sure about the toxicity study quality. And then 2,6-xylenol is also reported negative in NTP salmonella assays. So with this, I thought maybe we could look at it, whether this makes applying the carcinogenicity threshold, and then move on to the next and then see the non-cancer and see how it works out. So the chemical information is, this is by class 1, by Monroe assignment and tox tree, and it is actually important to look at the Kremer class assignment a little more carefully. We will talk about this later. And then there is a FDA CEDI database, the cumulative estimated daily intake. So I just took an estimated the intake value that it might be that we are exposed at a certain exposure. And this compound is in cosmetics used as a perfuming agent. So we'll take this compound and go to next. first, we could not find 2,6-silent or cancer endpoint data. So we would be looking at both. Now, we're going to assume that this particular product case has a use level of this compound at 1 microgram per day. So then we look at, well, I don't really have any outstanding structural alerts for this. And we also know it does not hit real the genotoxic alerts, very outstanding alerts. And we already know that it's known to be Ames negative by NTP. So we're just going to assume that it may not be mutagenic. So then, since the EDI of that cancer endpoint is 1.5 microgram per day, we are already below that. So we might be at this point, we'll be fine for this compound. It could be a negligible human risk if we were to be under that 1.5. Now, what if we decide to go look at further down the tree, and we're just going to follow how to use this TCC decision tree. So the first thing it'll ask after that gene tox, so you will go into the green region, and the first question number there is it organophosphate? And obviously it is not. Then we proceed to the next. Then it's going to actually assign the CREMA class. So is it CREMA class 3, 2, or 1? So here's a 3, 3, 2, and then here's the bottom, CREMA class 1. And if it's a CREMA class 1, and did you have a question?
No.
Okay. The compound is CREMA class 1, and the EDI, the estimated daily intake, is definitely not greater than 1,800 microgram per day. So it will be both safe, either cancer or non-cancer in this case. Safe meaning the negligible human risk can be assumed. So it's a pretty, assuming that this was a good case, good example. I'm not saying that it was difficult to find an example, and this is how we would normally use a TTC tree. So then I'm going to move on to the next topic of the cosmetic-related chemical and its chemical space. How do I know that certain chemicals are cosmetics if I'm not working in a cosmetics industry? And how do we actually apply this? So that's the point. The next thing is, the reason that we have to understand this chemical space is because of the applicability domain of our database. The two requirements of any TTC approach, actually, whether it's a cancer or non-cancer, is we need to know the chemical structure and understand the chemical space of that. And the other one is the toxicity, good solid toxicity data with NOAEL decision to come up with a pragmatic TTC approach. So the chemical class will be actually evaluated by defining the chemical space. And the reason this is important is because TTC concept originally was developed from oral exposure studies made primarily for food contact materials assessment, as you would remember. So in this case, there are several questions we have to ask. Is this applicable to cosmetics-related chemicals that are used in the cosmetics products? Also, does Monroe, the current database of Monroe, does it cover enough cosmetic ingredients and impurities found in the cosmetic products? So this actually, some of this has been looked at earlier, and there was an important study by Karen Blackburn at P&G in 2005 that there was a feasibility study based on the small personal care products. The database wasn't large, but the way that it went about was to compare the chemical space, and it was a quite positive outcome that it may be possible, that it's probably feasible to extend. Then the Cosmos, and leading by Andrew Worth back into 2012, looked at this cosmetics chemicals, related chemicals, and how they could be related to the Monroe and others. So there was some studies done before. To provide a reference list, a lookup list for defining substances used in cosmetic products, what we have done in Cosmos is we established a cosmetic inventory combining both European and US list. So the European list came from the coding, and the US list came from the PCPC book and which is actually basically part of our the list from the Voluntary Cosmetics Regulation Program, the VCRP list. And now we have a collaboration with USCIR. They are actually helping us to update this regularly. So combine those, there are overlaps between those two, and not completely. There are about 100 compounds that we don't see in that we see in US that we don't see in EU, and there are some like that. Anyway, the Cosmos inventory, the cosmetics inventory, in total, about 20,000 inky names and about 1,000 CAS-RN. Why is it a one-to-one match? Is because there are many compounds that are substances that are not structurable, that are botanicals, and because of and also many-to-many relationships. So this has been a very actually difficult inventory to put together. We have done this finally, and we have implemented within the Cosmos database. So Cosmos database you are already familiar with through this webinar. So I'm only going to go over this the cosmetics inventory, where we have more than 17,000 inky names that we have actually registered, and some structures where we actually have curated and the QC under QC, and we have also annotated the coding chemical functions and the VCRP product category. And the special thanks acknowledgement goes to the FDA and DSS Tox and P&G, who have provided us the initial structures of this database. So just to show you a glimpse of this cosmetic inventory, that how it is implemented in the Cosmos DB, you will find a compound like zinc PTO. Once you search for it, then you will see the coding, the database number, and it could actually take you directly to the coding and SEC's opinions. If there is an opinion written and there's an inky name, all the names are actually annotated, including the inky names. And also the coding function, chemical functions, and you could click on that, and you'll be able to see the definitions of those chemical functions. And this is all available from the Cosmos database. Now, analyzing a bit of this cosmetic inventory these are the top 10, the chemical functions in the product category, the skin conditioning and protection, the emulsifiers, the perfuming, the hair dyes, the hair conditioning, the emollient, antioxidant, any microbials and preservatives and UV filters and plasticizers. You can see those very much that whatever it actually has is chemical functions and the product category that annotated with, it has been then known to be used with the cosmetic products. Then we take a look at this cosmetics inventory with the chemical thresholds in the chemical space by applying this different various what we call a chemotype. These are structural features, specific structural features that we categorize as compounds. You can see there are alcohols and phenols, and we list them over here. But again, certain chemical compound classes are populated, and there are a lot in the hair dyes And these are the ones, the ketones and ACAC and carboxylate esters. These are the perfume and fragrances. And then down here, the non-ionic surfactants and anionic and cationic. These are the emulsifiers and stabilizers and surfactants. So you can see actually very nice the categorization of these compound classes. And this is just to graphically show you how the cosmetics and food are similar and dissimilar. The cosmetics and food chemicals are actually, interestingly, very similar. This is my joke about the women eat about a kilogram of lipsticks through their lifetime, and what we eat is what we apply on the face. And many of the chemical classes are similar, but there are distinctions. We don't eat surfactants. And you can see right away that properties are different. And so this is a good picture to remember how the food and cosmetics broadly share the similar chemical space, but also different. So we have just covered the brief history of the TTC and chemical space, definitions of the cosmetics-related chemicals. So let's then move on to combining the two, our goal of the TTC approach for the cosmetics. Again, you have seen this before. There are two things we need. One is a chemistry, so we have defined the cosmetics inventory. The other is the toxicity data. And there are issues in this toxicity data availability. And some of them actually related to regulatory requirements, because there are only few cosmetic ingredients are subject to EU regulations, for example, coloring agents, preservatives, and UV filters. And so many of them have different levels of toxicity testing, depending on the application and substance types. So in result, repeated dose toxicity data are very difficult to come by for minor components and impurities. And some of them, especially if it's ingredients, and much of this data can be also proprietary, understandably. And the fact that regulators and people in the safety assessment need immediate availability of data doesn't change. So here comes the need for our database of NOAEL and NOAEL database of good data. Another issue of this applying this TTC approach to cosmetics is the fact that most of our good data, good toxicity data, were collected, the experimental data are in oral route of exposure. However, the cosmetics products, many of them are actually delivered dermally. That's the primary exposure route. So there are two ways to go about doing this. One is develop dermal TTC method based on a reliable dermal repeat dose toxicity data, if we have them. And the second route is to extend the oral TTC by applying an oral to dermal extrapolation approach. So when we first started this project, the Cosmos has a scientific advisory board, and Susan Felter was one of them, is one of them. And we had many discussions about this. And the fact that the dermal data, the good dermal data are harder to come by, we decided to take her recommendation of extend the oral TTC and then to address this oral to dermal extrapolation. And that was also somewhat on the feasibility studies by the Cross and others in 2007 was also recommended that, right? So we are looking at this, the way that we set up at Cosmos. Let's go back, okay. With Susan's recommendation, we have contacted the ILSI Europe who has been a leader in the TTC field, and we established 2 expert groups. So the first group is to come up with the oral TTC data set that we could use to extend this approach enriched with the cosmetics-related chemicals. The second group is to address the bioavailability and so that the oral to dermal extrapolation can be addressed. And then those 2 expert groups will be combined once we have finished the work and come up with one combined strategy. So during this part, the expert group 2's oral to dermal extrapolation is, we will not cover that. It's in the talk of its own. We will cover mostly on the expert group 1's activity and for that group on this establishing oral TTC data set enriched with the cosmetics-related chemicals. Now that we are actually establishing a new data set through a new NOAEL database, we had to come up with a curation strategy of this Cosmos TTC data set, especially because there weren't enough data available out there. So the first thing we did was we consolidated all the toxicity data publicly available out there. We identified them and tried to consolidate. However, we needed a clear study inclusion criteria so that we could filter. We could actually include first the good studies that we want that meet our criteria, and then we establish our NOAEL selection criteria to filter those studies into our NOAEL database. And then into this NOAEL database, we added Monroe 1996 data set. Then based on that, total together, then we apply for our further criteria of study relevance, the toxicological relevant and human relevant criteria. And this is where the ILSI toxicologists and through the expert group 1 have come up with, gave us guidance on this series of criteria so that we could select the right NOAEL values for the final TTC data set. And so far, based on last almost 3 years now, 7 iterations of this data set have been evaluated, and we have finished 2 full QC sessions of these studies, some of the selected studies. So in order to do that, we first established a new toxicity database that has more cosmetics-related chemicals that center around there. So the data, actually, original data are from NTP, or the FDA SERES, or the EU SCCS, or ECA, and also the literature. And we apply our criteria, and then we set out as what we call oRepeatTox database. And now this is actually implemented in the Cosmos DB. You will be able to find this when you search for Cosmos DB for a chemical. You will see this separate oRepeatTox database data presented to you. And they have been manually harvested over 2 years. And there are about 230 chemicals and about 100 hair dyes, because we have harvested SCCS data, and they have actually had many opinions on hair dyes. And then we also included some of the food additives, the contact substances, and then their impurities of the 42 impurities that provided by the SERES FDA SIPSEN. So total, there are about 340 oral toxicity studies on covering chronic subchronic, and only the subacute if it's greater than 28 days, and some reproductive developmental DART studies. So now I'm just showing you briefly that for a compound that first you will see that there is a oRepeatTox database. I'm not just talking about a name. It actually exists as a separate database in here in our Cosmos DB, and it will show you that our assignment for the Climich score, that this study was reliable with restrictions, which means that this was not a GLP study, but the design was good enough. And then it will list the dose levels and if there were any NOAEL values. So this particular case will show each dose level and the assays, whether the body weight was changed, clinical signs of what type of clinical signs was changed, and organ weight, and at different dose levels, and effects. It will describe the effects and the treatment relevance, whether it's a treatment related, not related, or statistically significant, and all those qualifiers of the effects. So what I just briefly showed you is that the result of what we have gone through to decide what is a good enough study for us. So the study inclusion criteria we actually used was just any familiar in toxicology studies, there's study types and what types of species has been used, what those levels were used, what kind of regimen was used, is it a 7-day or 5-day, okay, and how long was the study, and then each dose level we have an effect for the dose group, and was the reference actually tractable reference, and is publicly transparently shareable. And of course, in the center, all of our studies are oral studies. So these must meet means that we say rat, mouse, dog, monkey, and rabbit only for the DART studies. These are, it has to be one of those, and if it's not, we throw out. And must meet Cosmos minutes criteria, meaning the minimum criteria means that we do not take single dose studies, and how many number of animals that we need to use, and those are actually by the guidelines. And if it's not by the guidelines, and we actually have the number we say our minimum criteria. And again, over here in the duration, like I told you, that it has to be greater than 28 days, because the Monroe's approach was actually for the chronic. And so if it is any shorter than that, we throw out the study. And subchronic studies were actually, we were adjusted with a factor of 3 when we calculate the NOAELs. So through this process, I'm just showing you the profile of this oRepeatTox toxicity database. It's most sensitive species, of course, rat. There's a lot of rats showing up, and then the first one is liver, and then kidney are the most one. This is very common, and then the different types of lesions in liver, and the compound that it shows that you can see immediately certain compounds are the steatosis, and some of them are actually the pipa gamma related compounds. Trying to go to the next stage.
Is that what you're looking
Okay.
for?
Then, yes, thank you. Once we have a overall toxicity database, then we are actually have NOAEL values that we put together, and that's what we call NOAEL database. In that NOAEL database, all the compounds that have made our study inclusion criteria, and it will be actually populated the database. But when we take the next step of selecting for the Cosmos TTC dataset, then we apply the further set of criteria. So the first one is toxicity database. The inclusion criteria is the same. Study inclusion is the same as oRepeatTox DB. The NOAEL selection criteria is now very important for the TTC dataset. We actually went for the lowest NOAEL with clear LOL. And the freestanding NOAELs were excluded whenever possible. And we chose chronic studies whenever available over shorter term studies. And we preferred the guideline studies. So from the NOAEL database, we take some, applying this selection criteria of the NOAEL, and we have first a candidate for the data set. Then we look for distribution, and we found the lowest 10% of each Kramer class. Those compound records have been all QC'd by the ILSI expert groups. And also, because our data came from several different data sources, we look for the conflicts across the different data sources. And if they were different enough, we have an actually criteria for that. And if they are different, varying enough, then we include it in our review, the study review. So as a result, overall, it's about 25% of this database have been meticulously reviewed by our toxicologists by ILSI. And when they were looking for was, they applied the same criteria, the toxicologically meaningful and human relevance, and over any finding the lowest minimum value of the NOAEL. So this was a very labor-intensive work that they have done, but through this approach, we have learned very valuable process which we are applying, which can be applied consistently. So let me just briefly explain about this 1.7. The version 1.7 is still not totally completed. We just actually finished the last set of cleanup. And you can see this started from 1.0. So it has been going on for 7 different iterations. There are about 558 substances. When we first started, there were over 700. And we threw a little bit of studies and data. Of those 558, 500 of them are found in cosmetics inventory, and Monroe has about 180. So remember I told you that does Monroe have enough cosmetics chemicals in the database? And Monroe is amazing. It still has quite a good percentage of that, 180 over 600. That's not a negligible amount. And we look at the distribution of the Kramer classes 1, 2, and 3 between the Cosmos and Monroe. You can see Cosmos TTC dataset has more percentage-wise of the class 1 than the Monroe would have. Monroe is the other way around. Monroe has a lot of class 3. A couple of things that our experts and both toxicologists and chemists agreed on to act on is that we remove lipid-soluble vitamins A, D, E, and K from the database from the TTC data set, and then also the essential amino acids. In compound classes, by looking at the compound classes, we have quite a few hair dyes, 110. As I told you, 100 of them came from our manual harvesting of the SCCS. And then we have parabens and phthalates. And this is also a large class of its own. But those actually as a potentially in the database. We have not decided completely yet what our TTC version 1.8 will be, and it will be decided in the near future. So the first thing is we're going to compare the Monroe TTC data set with the Cosmos 1.7. And by looking at the chemical chemotypes, the alcohols and amines, the blue is the Cosmos and the Monroe is the, Monroe is orange, and then the gray is the cosmetics in Monroe. So if the length is about similar, then they are between the no difference between Cosmos and Monroe. If the one is only in one color, that means it only shows up in one thing. So for example, the steroid ring is only in Monroe, we don't have in Cosmos, and the sulfide is in Monroe but not in Cosmos, and phosphorus organogroups, a lot more in Monroe than in Cosmos. Now we have many hair dyes. So you could look at the ethylamines, for example. That's actually a real, it's a theme of a chemotype for being showing up in as in many old hair dyes. So you can see that we have a lot more in Cosmos than in Monroe. And what I'm showing you in the property-wise is the Monroe and Cosmos are also similar, but Cosmos is a lot more diverse because we have surfactants and other more diverse cosmetics chemicals in the database. Okay, there are still issues we are addressing right now, is that while we are doing this, we learned some valuable lessons that the expert assignment of a chroma classes and a software assignment are not always agreeing each other. So for this, this is a bigger issue than just Cosmos, but for right now for cosmetics TTC data set, we have been resolving this manually. So Steve Enoch and Kirk Gavison and myself, we sat down and just, we bite the bullet and did that. Okay, then the next one is there was still unreliable data within the 5 percentile that did not get reviewed because other issues, like for example, isopropyl alcohol, EFSA reviewed, but it turns out that the data was not available. And isopropyl alcohol, the Monroe 0.018 for class 1 is for isopropyl alcohol, and it is not a reasonable number. Another one was ascorbic acid, for example, ascorbic acid L and ascorbic acid, and the difference between the two was several thousand-fold, and again, it was not an acceptable variation. Another one is 4-butyl and lactone, and these compounds were actually driving also class 1 in unnecessarily low. So we have done another review on this one, just particular for these three, and we just finished this. So we'll be actually presented to our expert groups. Then we have another story about the parabens and phthalates, because Cosmos found quite a bit of these compounds in our data set. And Monroe, back in 1996, it's a 20-year older studies. In general, they were less conservative than Cosmos NOAEL database. But then again, this was there were a lot of actually newer studies available. So at this point, what we're thinking is if the compound is really well known and a lot of studies are available, and how do we handle this? So because data TTC is actually established for the compounds whose data are not known or limited. So anyway, so our expert group is now currently evaluating how to handle this. Then we have also evaluated the inclusion of hair dyes or the not including. And it turns out that whether we remove or not remove separately, isolating out, it didn't really make any difference in the 5th percentile values. So we decided to just leave it in there. And then the Cramer class 2, we all know that this is not a viable group, not enough statistically. So we are going to evaluate actually to see whether we could combine with the class 3. So I'm actually at the end, I made it through this. The next step we have planned is that we will finalize the Cosmos TTC dataset hopefully by end of this fall. And there may be some more studies that we need to review. And so our expert group is actually considering that right now. Then another one is that we will also finalize the skin permeability database that put together to support that expert group too. And we haven't actually put neither of this database into the Cosmos database, and this will happen once we all agree and clean up the data and then import it into the database. And after that, it will be publicly available with the Cosmos schedule. Then what we are also planning on right now is establishing this, developing TTC tools. So you could see how I just went through a very simple example of you have a query compound, you assign a Cramer class and other compound categories, and then check the chemical applicability domain compared to the database. Then we could apply this TTC decision tree. And another thing that our expert group 2 has done that I have not gone over is that including the bioavailability, especially the exposure part, into this TTC tree so that we could address the oral to dermal extrapolation. So overall, this is the next slide. Christy, can you get me the next slide? The acknowledgement.
Okay.
So these are very important acknowledgements that we actually owe quite a bit of gratitude to our expert group 1 and 2 of the ILSI. And then our Cosmos partners. So you could see that some of the Cosmos partners were also in the ILSI expert group, but the main people in the expert group in the ILSI for the PTC database was Alan Gubi, Susan Felter, Helge Holnagel, and Christy Jacobs. And the US EPA for sharing the ToxRef database with us, and the CIR for providing us its VCRP list, and updating us regularly. And Sips and Sarah's team, especially Kirk Arvidsson, and their donation of their structures and the initial Sarah's DB, the public part, and then P&G, they have given us 30,000 structures in the beginning. So very important contributions. And at the end, we have to mention our funding sponsors of European Commission and Cosmetic Europe, and we hope to provide you some data in the near future.
Well, bravo, Chihae. Thank you very much for that presentation. I think I'd like to apologize for the trouble with the slides. I'm not sure, that our system just wasn't displaying them correctly, I guess. So We're over time, obviously, but lots of you are still sticking around. So I'm going to open up the floor for questions. I'm not going to unmute everybody because it's the background noise is very loud. So what I'm going to do is ask you, if you have a question, to type it into the chat box and send it either to the organizer or to all is fine too. We have one question already. It is from Bola Taiwo, and it's: suppose you use two toxicity prediction tools, for example, ToxTree and a different tool, to determine the category of a chemical, and one method indicates the chemical has a structural alert for mutagenicity, and the other shows that the chemical does not have an alert for mutagenicity. Which result should one believe, or what further step should one consider? I think is probably the key question.
Yes. So I'm not on. I could talk, right? I think this study is one of the most difficult, actually, aspect of computational toxicology. Is that so let me say, provide some explanations that why there may be difference in opinions. The structural alerts can be, actually, the levels are different. Some of the structural alerts, if I say aromatic amines, are all genotoxic, that would be, I would run into many false positives. I would run into trouble. But in general, if I have aromatic amines, people also would understand, oh, you got to be careful, right? So it really is up to how you represent, how you want to be specifically represent that structural alerts in terms of, representation, whether it is a SMARTs or chemotypes, whatever we use. And because of that, what we need to provide really, we have an obligation, I believe, the computational people have an obligation to provide a likelihood values for each of that alert. How likely is it really true, right? So if I say aromatic amines, a specific aromatic amines, and with the, also empty, or, certain, it's a primary aromatic amine and also positions empty. If I could say things like that, my likelihood will be different than tertiary aromatic amines. And we need to provide those likelihood values, then we should be able to compare them better.
Great. I think
so that's, I know in ToxTree, so Bonini-Bossa rules will have certain, will say certain things. And then the also, famous product like Lhasa would say, they have a knowledge base and they would say these things. So what you have to ask at that point is really how likely is it? The likelihood value. Odds, what we use is the odds ratio.
Okay. We have another question about Cosmos, wondering about when the TTC will be available, and when will the bioavailability tree be available?
Okay, so the bioavailability tree, that manuscript is now being put together. We're actually at the final editing stage.
Okay. And that's for dermal bioavailability, is that right?
It's actually oral to dermal extrapolation related bioavailability.
Okay.
The last, I just show a last picture in my next step. There was a small picture. And that decision tree actually includes that our approach, how we go about the oral to dermal, when we use oral, when we don't use oral, and what the factor maybe we should think about. And these are the things that the decision tree goes about. Yep. And that will be published soon. Faith Williams, is actually trying to push this manuscript soon.
Mm-hmm.
Okay, so that, and then the skin permeability database will be also included in the Cosmos DB. So we're thinking by end of 2014, that part. Okay. In terms of the Cosmos TTC data set, I just like to update you guys that this is each QC work that we have done is a long process. So first of all, getting ready and for the experts to actually review, and then we receive and we collect, and then we actually do another type of cleanup and then process this back into the database. So that's what the last cycle went through. This cycle usually goes through longer than 6 to 9 months after the QC has been done. So now we have only some issues left we are still discussing. We are hoping that we'll get this done sometime this fall, and then the ILSI Expert Group will be actually evaluating this, and then after that, once it's imported to the Cosmos DB, then you'd be considered that as a public release.
Okay.
Did I answer the question? So I'm saying it'll be after end of this year.
Okay. Yeah.
Okay.
Yeah, definitely. Jeannie asks, why were the amino acids removed from the database?
Oh, only the biologically active, the real biologically active ones. Because the TTC approach is really not used for the essential amino acids, the really essential food amino acids, and then also also the human, whether the human endpoints are known. I mean, so it wasn't really. There was a good reason why we removed it. Those are the reasons. That first, we don't apply TTC for essential amino acids, essential to the human.
Mm-hmm. We have 2 questions about basically asking about sort of official acceptance by regulatory agencies. In other words, are you able to use TTC for safety assessment of cosmetics in a regulatory context? And I think that's somewhat outside of the scope, but if you want to say a couple of words.
I really, I'm not from any regulatory agency, so I cannot answer that.
Yeah.
All I can say is that my colleagues, the professional colleagues I know, they would use it if the method is acceptable, if the method is clearly, transparently documented and solid, right? Mm-hmm. So it's our responsibility to provide a solid method first.
Yeah.
So we are working on that.
Great.
Mm-hmm.
And there's been a couple of requests for the slides. I don't want to put you on the spot, but it's possible that they'll be available simply on the Cosmos website or the AICCT website. And
of course.
Yeah. Okay.
Yeah. I just hope that people understand that my examples might not have been the best one. Okay. It was hard to find the right.
I'm sure people can understand that. Well, we don't have any more questions, so I just want to thank you, Chihae Yang and Andrea Richards, for talking to us today. There's clearly been a lot of work by your team, and there's clearly a lot of excitement and interest in this update of this useful tool. Anyone still listening, you can again, I'll point you to the Cosmos website, which is cosmostox.eu. That's cosmostox.eu. And also for more information on the ASCCT, you can go to ASCCTOX, ascctox.org. And thanks to everyone for your attention. And we'll end the webinar now. Thank you, Chihae.
And thank you, everyone.